Sorry for the technical hitch. So thank you very much for the introduction. It's a great pleasure to give this opportunity, to be given this opportunity to talk this afternoon. I'm Olivier Hanotte, and I have two hats. As mentioned, I work as a professor of genetics at the University of Nottingham, in the UK, but I'm also a scientist at the International Livestock Research Institute in Addis Ababa, Ethiopia, and I'm actually talking from there today. What I'm going to talk about today is actually a work done in collaboration with scientists in Korea, and in particular Professor Ibal Kim from the University of Seoul. It's a long-standing collaboration. We have been collaborating together for the last 5 years. The starting point, of course, of my presentation is cattle, but the enormous, and I would say the huge diversity of cattle that we do find on the African continent. Here are some examples. You do have, for example, the Dama cattle, which live in West Africa. This quite extraordinary white animal with huge horn are called curry cattle. They live around Lake Chad. This is the Watusi cattle, which are related to the Ankole cattle, the famous Ankole cattle from Uganda. Another example here will be this bull, which is a Fogera cattle living around Lake Tana in Ethiopia, and so on. So you do find on the continent a huge diversity, huge phenotypic diversity of cattle. And obviously that triggers some question. What is basically the origin of this diversity, and where does it coming from? And if you are interested by these two questions, well, you can rely on two types of information. Archaeological information and, as I will show later, genetic information. So what does the archaeology tell us? Well, if you are fortunate enough to go in the Sahara and to look to the rock painting of the Sahara, what you will find out is this kind of picture, like in Algeria, desert, where you do clearly see animals which are cattle. These animals are humpless. They do not have a horn, hump. Sorry, they do have a horn, but they don't have a hump, and they do have a diversity of coat color, including, for example, this one, which is very much will remind us of the coat color of the Holstein Friesian. Now, if you move to Egypt, you equally find some iconography of cattle. But the point I want to make here is this animal here, where you can see this animal has a hump. So here we have Toran cattle without a hump. Here we have an animal has a hump. And it's one of the first evidence of a humped cattle on the African continent. In other words, of a zebu type or indecent types of animals. So we have been studying African cattle for many, many years. And the first main study that we published was in 2002, nearly 20 years ago. And at the time, we were not relying on genome analysis, but on other marker, microsatellite loci marker. And our study included 50 indigenous African cattle breeds from 22 African countries and a large number of samples. And combining the outcome of this study with archaeological information, we were able to propose, if you want, the timetable of arrival on the African continent of cattle. Now, the point to stress here is that today we do not have any evidence of a domestication center for African cattle on the continent. All of African cattle are coming from outside, and from the two recognized domestication centers of cattle, the one in the Near East, where the Toran cattle originated from, and the one from the northern part of the Indian subcontinent, where the zebu cattle come from. And this animal from these two centers penetrate, if you enter the African continent, in different ways, illustrated here in different color, and then disperse all across the continent. One thing which we didn't know, and which will be addressed later in the talk, is basically when did these cattle actually move from the entry point to the southern part of the continent. The archaeological information are scattered and still unclear. So this slide is making 2 points. The first one is the today geographic distribution of the different type of cattle on the African continent, blue being the zebu type, brown being the Toran type with the region of crossing, and green being what we call the Sangha cattle in Africa, is corresponding to ancient crossbreeding. And on the left, what we do have here is to illustrate the fact that compared, for example, to the Eurasian continent, the African continent has a north-south orientation rather than east-west. And of course, this is quite important if we think about agroecology. And you do find on the African continent a set of a diversity of agroecology because of this geographic orientation. So not only these cattle arrived from outside in different ways, but they were exposed on arrival, of course, to a diversity of environmental challenge. And so we became interested to understand if this environmental challenge would have left imprints in the genome of these African cattle. So we published the first piece of work 4 years ago in 2017. This was actually, to the best of our knowledge, the first time that the full genome sequence of African cattle had been studied. At the time, it only included 48 genomes from 5 indigenous breeds, and we were able to identify in the genome of this animal a set of signature of positive selection, candidate signature of positive selection, and which overlap with gene related to tropical adaptation of this animal. And the one I want to point out in this talk is a signature selection about the prolactin hormone gene. And it is not known that the prolactin pathway is involved in thermoregulation, as well actually in milk production, and also in the coat structure, actually. Okay, so archaeology is telling us about the early arrival of African pastoralism in the north and the whole of Africa, and also little information is suggesting the late arrival below the equator. Genetics is telling us that African cattle are not indigenous, that the majority of the animal are unmixed zebu taurine, so unmixed cattle with a ham and cattle without a ham, and multiple wave of arrival, and genetic genomics allow us to identify candidate of signature of selection in the genome of cattle following this arrival, corresponding to adaptation to environmental challenge. Now, what about the link between the archaeology and genetics? Well, how can we make this link? And in fact, this is the story which we are actually showing in a more recent publication in 2020, where we analyze a much larger number of African cattle genome. And basically, this is what the novelty we have in the Nature Genetic paper. So in the genome biology, we characterize this admixture of the taurine, and we did some signature of selection analysis. In the Nature Genetic paper, we actually date the admixture, and we also tackled the issue, tackled the issue of the origin of this admixture. So we did admixture, but also for the signature of selection, what are actually the ancestry of this signature of selection? Okay, next slide. Next problem. Can you see? Okay. So, can you see the slide now? Okay. So what we did in the Nature Genetic paper is actually we studied much large number of genome. We had actually 45 cattle breeds, including 16 indigenous African population and corresponding to 172 animals, and quite a large number of genome sequence. And the first thing we do when you get that kind of information is to look to the population structure and admixture, characteristic of the indigenous African cattle in this case. And what you can see, if you assume 3 ancestral background, is that in fact the African cattle, the East African cattle in particular here, do have an admixture between the taurine and the zebu, so the pink and the blue. It's only the African taurine which actually have remained nearly pure of zebu introgression. Now, we apply different approach for the dating of this admixture. And this is illustrated on this slide. And what we actually, the result indicated that if we take the cattle from the eastern part of the African continent, with the exception of the Kenyan Boran, and I can talk about that later if anyone is interested, but the admixture seems to have taken place around 150 generations ago, and which correspond, if we do a cattle generation time of 5 to 7 years, it corresponds to 750 to 1050 years ago. So what our results show and what our results are able to date is a major admixture event occurring approximately, let's say, 1000 years ago on the eastern part of the African continent. Because of the limitation of the analysis, we cannot exclude that some Zebu Taurine mixture occur early, but clearly there was a major one which occurred around that time. Then the next things we did is actually to look for the signature of selection identified in this cattle, if they were actually predominantly of Zebu or Indocene origin, or if they were actually of Taurine origin instead, or a mix of both. And what we discover is that, in fact, quite a large number of this signature of selection analysis, this candidate positive signature of selection analysis, were of a specific background. So the ancestry were not random here, and they were either from Indocene or either from Taurine. It's an example here on the chromosome BTA7 of cattle, where we have an excess of Indocene ancestry corresponding, overlapping with genes which are disease-related genes. We have another example here, actually, which is an example of taurine excess ancestry that correspond to actually in a region where we believe there might be a regulatory region for a protein, CAT11, which is might be involved in the tolerance displayed by some African cattle, and more notably taurine African cattle for sleeping sickness trypanosomiasis. So basically now combining admixture characterization, admixture dating, signature of selection analysis, and signature of selection ancestry, we believe that we have now been able to actually link history and genetics, and that in fact this, if you want, area of the crossroad of the different waves of arrival of cattle on the African continent, a major event occurred 750 or 1050 years ago, and this is where actually following these events, the cattle, which the main consequence of the event, if you want, was that suddenly you have enhanced the diversity of the local population, and this enhanced diversity combining taurine and Zebu, we believe is that what has allowed cattle to actually migrate it further south and to colonize new habitat. So the last point of my talk is that we believe that the future of cattle production in Africa is in these ancient indigenous hybrids. They carry important adaptation for environmental adaptation, which will be key to the improvement of productivity in the future. Thank you very much.